All right. So very happy to be back here at this conference, and thanks to the organizers for allowing us to take you through the VectorY story. So let me take you through VectorY's journey in science and taking antibodies. This is our tagline: taking antibodies where they have never been before. So very excited about that. We have a senior leadership team with exemplary experience in the gene therapy field with PharmaCell, and also strong legacy from Unicure, one of the pioneers in this field, so executing on our strategy. And VectorY is all about developing a broadly applicable, versatile platform to enable a new class of therapeutics to achieve targeted protein degradation. We focus on proteinopathies to treat neurodegenerative and neuromuscular diseases by combining antibody and gene therapy technology to address key therapeutic challenges. And let me take you through what these are: about efficacy, delivery, and manufacturability. So efficacy, to get to the right target, binding it, and achieving protein degradation. Getting to the right types of tissues and cells with the right type of capsids, and also manufacturing it at an attractive cost of goods sold, and also being scalable. So we employ synergistic technologies in our company altogether. Targeted protein degradation on VecTabs and Vectrons. I will take you through what they are. Our tissue-specific AAVs, capsids that we're developing based on AAV5 platform, and we're working hard on developing our scalable CMC platform, proprietary technology that we're developing on a baculovirus insect cell-based platform. Combined all three of those into what we believe is going to emerge as a new class of therapeutics, where each one of these technologies alone, we believe, has an attractive potential. Our VecTabs and Vectrons can achieve target protein degradation, basically are a biological equivalent of the PROTACs. We are using a structural engineering approach, our VCaps, to improve the tropism for specific capsids, and we'll show data on where we've already been able to demonstrate that, and we're working on our scalable CMC platform on the baculovirus insect cells. So not only in CNS, where VectorY is focusing, we see application of the technologies in a broad range of other potential indications: immunology, oncology, etc. Also on the right top there, we have already expressed increased transduction compared to AAV9 in muscle and blood-brain barrier, so very excited about that. And also our AAV gene therapy platform will prove to be versatile to really bring these products forward into the clinic. So what are the technologies that VectorY is employing? So our VecTabs are basically a precision targeting single-chain FVs or full antibody expressed, where we focus on the misfolded proteins and the aggregates where the wild type remains intact, and that's very critical. That's exemplified here in this cartoon. Our Vectrons can augment the VecTabs by fusion proteins, by offering pathway-specific proteolytic removal of these misfolded protein and aggregates. So there are three pathways: UPS, chaperone-mediated autophagy, and general autophagy, and we have our tools can address specifically each one of them individually. Our VCaps, we're using a structural engineered AAV5 capsid, where we believe will have broad application in both the muscle, but also blood-brain barrier and cell-specific tissues. And our scalable CMC, we're taking that through to a very carefully industrial design exercise before we scale it up. Okay, where we go? Yeah, next. So our pipeline, as we see it emerging here, is really going to be extremely differentiated. We'll talk you through our two lead programs on the OXPC and on TDP-43, two phenomenon that play a key role in ALS, where we see an IND coming in the latter part of 2024, where we are the first taking this in the CNS, but also in TDP-43, an even more validated target. We are seeing intracellular expression of that and leaving the wild type intact, and show you data on reducing protein aggregates already in our initial experiments. And then furthermore, where you look at the platforms, the vectrons and also the capsids, we tend to apply them in the polyQ Huntington area and alpha-synuclein to combine both the aggregate degradation strength of our platform, as well as the delivery aspect to those pieces of our pipeline. So all of these technologies are playing together to achieve the optimal therapy. So, okay, here we go. Yes, so on our VecTabs and Vectrons, so getting to the hallmarks of ALS pathology, and we see that as a proof of concept in an orphan drug disease. So let me take you through the pathology of ALS real quick. So two important phenomena play a role: the protein misfolding that actually leads to then the toxic protein TDP-43 aggregates. We express them as single-chain FVs, but the TDP-43 aggregates are toxic and lead to mitochondrial dysfunction and lead to increased oxygen radicals that then lead to this cascade of oxidized phospholipids, the neuroinflammation, and ultimately cell death. At the same time, you see in ALS patients an increased phospholipid synthesis that leads to actually a buildup, because the neurons have low capacity to metabolize them, and this buildup then leads to further this cascade. So these two phenomena come together, and we address them individually through our combined platform. This is a picture of a neuron. Actually, ALS pathology builds from the neuromuscular junction, and here you see that in fact the translation cytoplasmically from these aggregates leads to dysfunctional translation in the mitochondria, leads to rapid reactive oxygen species being formed and causing damage here in the neuromuscular junction. And we're addressing the aggregates here. At the same time, this other phenomenon that talked about phospholipid buildup will lead to oxidation of the phospholipid head group, the oxidized phosphocholine group, that then in the membrane, this is a membrane here, leads to new epitopes that cause downstream inflammation in ALS patients and to neuronal damage, as I just described. And we have an antibody, single-chain FV expressed, that actually blocks that inflammation process. So how does that work? We're using AAV5 vector, have the transgene built in that is specific for the aggregates, leaves the wild type alone, and as a result will hopefully improve the disease pathology. And I'll come to the right-hand side of this slide right after. And we, in fact, the only company now really addressing virtually all of the ALS patients, because this is actually expressed in over 95% of the patients, whereas a lot of the other alternative strategies employed are actually only addressing a small minority. The key here, for your information, is that the nuclear TDP-43 is what you want, and it's critical for cellular function. The cytoplasmic TDP-43, the phosphorylated TDP-43 that form the aggregates, are actually the toxic ones, and those are the ones that we are targeting. So here we show the number of experiments we've done, and I'll come back to this. Number 51, you see this is actually the aggregate expression in the cell. It's actually here. This is normalized. We have a reference here. Number 51, I'll come back to that, actually shows aggregate binding but no reduction. But here on the left-hand side, we did a number of epitopes screening there that the VecTabs here show a remarkable protein degradation. So we're very happy with that, and it's actually specific for the aggregates and not the wild type, as I said before. So this is a rapid experiment to show that indeed, with the AAV5 single-chain FV, we can achieve the whole cascade of expressing it in the getting biodistribution in the key tissue areas, have mRNA expression, good correlation with the vector DNA as well as with the single-chain FV, and also we have protein data that show that we can achieve therapeutic level concentrations. So this is a pig experiment, so a large animal. So in fact, we can transport these across a long distance. On the OXPC program here on the right-hand side, you see again AAV5 transgene now expressed as a secreted antibody here that attacks the neoepitopes that are forming on the membrane, and in that sense want to reduce the inflammation. And it's demonstrated here that the OXPCs, the oxidized phosphocholine, are present in the motor neurons here. In the healthy cells, you see just a normal picture. Here, this is the EO6 mouse monoclonal antibody that is used as a marker, you see actually being tied exactly to these motor neurons. So this is where the inflammation is, and this is where we aim our therapy to go. And we demonstrate that here with the antibody in this experiment. You challenge the cells here with OXPC increasing dose and measure the neuroid outgrowth. And actually you see that the neuroid outgrowth is less, so it's toxic. We use the EO6 antibody, and in fact we block that effect. And we see here in the picture a normal dendrite formation, whereas in the control, those are heavily damaged. So same experiment here, but now in the mouse. Again, we can show here that we actually can achieve biodistribution, mRNA expression, as well as a good correlation, so for the expression of the single-chain FV and also the protein expression. So what's next? So next is what we can do better than just with the VecTAPs, and we can. Enabling pathway-specific protein degradation. So how do we do that? So we know that there are three areas that are very well in focus: ubiquitin proteasome system, chaperone-mediated autophagy, and autophagy. So what we did, we attached fusion proteins to the single-chain FVs to see whether we can direct them specifically to any one single of these pathways. And this is the experiment. Remember the 51 I mentioned? This is our reference. It binds to the aggregates, but it doesn't degrade them. And here in the color scheme, you see the three different pathways. And here all the way on the left, we can show that, in fact, for all three different pathways, we can have a remarkable reduction in protein clearance. And so not only do we bind and stop the formation of these proteins, we actually clear them from the cell. So that's rather unique in our approach. So moving on to our further technology to enable future therapies, our VCAPs, the structural engineered tissue-specific AAV5 capsids that we are using, then we believe that AAV5 is a really step forward in our next generation gene therapies. We've developed a AAV5 proprietary vector that we believe has improved immunogenicity, toxicity, and CNS biodistribution, specifically compared to AAV9, which is generally used in the industry. And there's clinical data out there that support that phenomenon in AAV5. So that's where the basis we're working from. And here we see two experiments. This one on muscle targeting capsid in like these organ models that we have in our labs, where we show here this is our proprietary standard base capsid. This is standard industrial AAV5 with which currently clinical trials are ongoing. And we can see here the remarkable increase in transduction and expression here in these muscle cells with this experiment we did with really a multiple compared to AAV9 in terms of the expression of GFP in this case. Similarly, we did an experiment with organ on a chip in the blood-brain barrier with very tight junction here, and we see transduction here from the blood side over to the brain side with the base, but also specifically with our engineered capsid, strongly enhanced. And here are some of the data where we compare that, the AAV9 here, and then our improved capsid. So we're very positive about this technology and our ability to structurally engineer future capsids, not only for the muscle and blood-brain barrier, but also for heart and other, and the CNS cell-specific. And remember, we are implying to develop that also for our deep brain for Huntington and our alpha-syn program going forward. So finally, on our proprietary manufacturing, we've done really a bottoms-up industrial design effort to establish our baculovirus AAV insect cell program, and we now have a 2-liter platform that we can scale up to 50-liter. So that's really going to deliver us a scalable, stable, and also ultimately lower cost of goods, specifically for future larger indications. And we are establishing and have established a collaboration for CDMO with a CDMO in which VectorY will aim to have control over our GMP manufacturing as well. So we'll operate one section of this rather substantial building, and so we actually have control over the total supply line from both the process know-how as well as the GMP scheduling for future clinical trials. So we have not a virtual company. We're close to 50 people already, 1.5 years into operation, based at the Amsterdam Science Park. And we are now in 2022 moving forward with further preclinical work on biodistribution studies and functional studies on our lead programs, getting ready for IND preparation and the trajectory towards that, solidifying pipeline and platform, because what you've seen here is just the first set of experiments, and then we're launching also a financing round shortly. So all in all, I think quite an exciting proposition, and our team has been working hard over the last year to have these results, and we look forward to showing you more in subsequent conferences. Thank you very much for your attention. Thank you.